Okay, so part three of the set of notes and the rest of this chapter deals with DNA, and you can see some pictures here of models of DNA, of animations or whatever pictures of DNA. DNA is so small that we really don't have actual pictures of this. We have things like you can see in this picture right here. This is what we call a X-ray crystallography picture. So it's taking a picture kind of cross section at this angle right here through this. So you can see where, the twist occurs and stuff like this. This was the picture that Watson and Crick that you see over here in this picture of these two men were able to use as part of their discovery of what DNA actually looks like. And then we have these pictures of bacterial DNA down in here. DNA is genetic material, so DNA has to code for things. And so the rest of, besides, just the general structure of DNA, we will get into its actual function, its actual job. What is it doing? Remember, structure determines function. So let's get some background in some terms and in some things that you'll need to know. First of all, remember that DNA, RNA, any of those, the Na stands for that group of organic compounds called nucleic acids. So it is a nucleic acid. And DNA is a code. But what is it coding for? It's coding for the production of proteins. So making the proteins is going to be what we consider to be our traits. So, hair color, skin color, eye color, those are physical traits that we see, but it's actually very, very small amount of your DNA that is coded for those sorts of things. DNA codes for every single trait, and most of the traits are things that will keep your cells alive. So they're exactly the same from one individual to the next. We are very, very, very much alike because as human beings, as mammals, as vertebrates, as animals in general, the things that keep our cells alive are the things that keep all cells alive, or at least within, this family relationship, this group in here. So really what we see as far as our physical traits is such a very, very small amount of our DNA. For instance, let me give you some percentages. And you got to think with percentages, you're talking about 100% of the entire thing. There's a lot of DNA. I'm going to tell you how many base pairs in a little while, but we are about 98% the same as like a chimpanzee. So 98% of our DNA that would be similar to a chimpanzee is not about our physical traits that we think of as our traits. It's about your cellular traits, the things that will keep the cell alive. We're about 92% the same as a rabbit. Okay. So that tells you how much of your DNA actually codes for the cellular level, not these physical traits that are the obvious things that we see, but the things that are required for the life of that cell. All right. So job of the DNA is to hold the genetic code, and then that genetic code will have to be translated into some sort of protein that will actually affect the functioning of the cell. All right, so let's again look at some terms in here. Genetic information. This is just the instructions on how we're going to make a protein. So DNA holds a genetic information. DNA doesn't actually make the protein. It's just like the recipe book, okay? The recipe book that holds the instructions on how to make different recipes or different proteins out of this. Then we can divide it down. So you have, as a human being, we have 46 chromosomes, okay? We have, if you want to look at it this way, we have 23 pairs. And we have these chromosomes in pairs because you get one copy of each chromosome from each parent, and they're exactly alike. So if I have a gene on one chromosome for, let's go with something obvious like my hair color, right? Then I have a gene on another chromosome exactly like it in the exact same place for hair color that I got from my other parent. So I have these matching sets of chromosomes. Each chromosome is a long strand of DNA that has many, many, many genes on it. you have 23 pairs of chromosomes, right? And on those 23 pairs of chromosomes, we have about 27, that this number changes. So it went from being 100,000 in the early 2000s, different types of genes. And then we changed that number as we learned more about our DNA. And we said, okay, it's probably about 30,000. And it keeps dropping. So somewhere within the range of we'll go with 25,000 genes. It's a little bit less than that because we're finding out more how they work. But so we have 25,000 genes, though, on just these 23 pairs of chromosomes. So each chromosome is divided into sections that we call a gene. Okay, so each of these would be a gene. So you got one gene, another gene, another gene. And each gene itself codes for one protein. Okay, generally. Our genome, all right, all these begin with the same words because it all refers back to genetic, but genome is your complete set of genes. So we have, your personal genome that defines you, most of which is going to be exactly the same as every other person's DNA. And then we have individual sets that define you as being you, right? So you're going to be different from any other individual on this planet, including mothers, fathers, brothers, sisters, or whatever. You're still unique. The only difference there would be identical twins. Identical twins actually share the same set of genes exactly because they came from one egg and one sperm, all right? But we can define this. We can say the human genome. So the human genome has all this complete set of genes that we all have. We just don't share them exactly alike. So brown hair versus blonde hair, it's still a gene for hair. So the trait, what the gene codes for is hair color, right? And there's actually a couple, but we'll simplify it. But the expression of it may be brown or blonde or something of that sort, right? So we have these traits. Every single human being has a gene for hair color. Now, what they express, what form of that gene they got, it may be a little bit different, but it still goes back to the same gene. It's just a little bit of difference in the coding, all right? Then we have this term genetic code. Genetic code is how we're going to go about from taking the instructions, right? The instructions, which is your gene, and making that into a protein. In other words, let's go back to that reference I made to a recipe book. How am I going to take, I've got the recipe book, which is your whole genome, right? I take one recipe that's held in the DNA, and I have to take that recipe and I have to make cookies out of that, right? So I have to take that code, those set of instructions, and then I have to make something out of that. That's what your body is doing. DNA holds that code, it holds that recipe, but other workers have to come in to actually make something out of just those words, right? Just that key to what it has to be. And that's what your genetic code does. It's how do we go about taking that code and making something useful for the cell? That would be proteins. So proteins, just think of proteins as being the same as a trait, okay? Because your expression, your trait, is actually a set of proteins that you've made. All right, we have been using genetic fingerprinting and stuff for a long time in all kinds of things, trying to find out, who's daddy, who's relationships, 23andMe, or any of those, so familial relationships. But we've also found that it's been very useful in different cases that have occurred in finding criminals and stuff, people who've done the job. The first case was actually way back in 1987. We weren't really all that good at it at that point, but we had enough information that we could begin this process of actually looking at DNA and linking an individual by their DNA to something. Tommy Lee Andrews in the United States was the first person to be convicted off of DNA evidence. And what he left behind was, a little bit of himself, basically, during a rape. When you look at DNA, we look at this in some sort of it could be a forensic case, it could be a criminal case, it could be identifying somebody from their remains. There was a whole bunch of DNA that was identified after the towers went down and stuff in New York. So there, it can be used for multiple different things, not just criminal cases. But in this case, this was the first criminal case that we had here. You always see something that's called the standard. Okay, the standard is just basically a known. We have to have, in every good science experiment, you always have to have a control, and basically you have to know if what you're doing is actually working and it has known elements about it. So a standard is a set of DNA, and you can see these little bumps right here, that has these stopping points that are known pieces of links of DNA. So they already know what the code is on this standard, and they're just going to use it to compare everything else so that we know what's going on with the rest of it. What we're really interested in is the sample from the person that you're trying to convict in this case, and so who perpetrated. We know we got this. We got that from the victim, and then the suspect. And you have to compare here, the sample that you retrieved from the crime scene with the suspect to try and figure out if that's the same thing. So here, when they ran the test, it doesn't matter the amount that landed in this spot. See how there's a lot there, and this has less. That doesn't matter. What matters is the location. So these two line up. So with that first analysis of that one section of DNA, they don't do all of the DNA. They take one little section. Because remember, the majority of your DNA is going to be just like everybody else's. So we don't really want to compare that. What we want to do is compare the portion of DNA that is unique to this individual. So they find that section and they cut that section of DNA out, and that's what they compare. But you don't want to wrongfully convict anybody, so you check more than one spot. You don't go with just one spot and say, hey, this is the person. We do a couple spots. So they use one set of DNA for the first section, and then they use a second set of DNA or section of DNA for the next. And in both cases, the crime scene DNA and the suspect's DNA that they took from his blood lined up in the exact same spots. And you would, nowadays we do a lot more analysis because we have the ability to do that. But this was able to convict him of that. It's much better than using somebody's, visual, what they remember from the scene, and eyewitness accounts and stuff like that, because those tend to under periods of stress, those aren't going to be as reliable necessarily. We can also use it for exonerating individuals. This is a tragic case here. Josiah Sutton was a case, I believe here in Houston actually, where he was convicted. And again, he was convicted off of eyewitness testimony, which, like I said, can be unreliable when you're under that amount of stress. Sometimes you misidentify people. It was in Houston. There we go. Houston Crime Lab. But they took eyewitness testimony and they used that during the trial. And then they looked at sections of his DNA. And the Houston Crime Lab, this was back in the early 2000s, the Houston Crime Lab said when they analyzed the DNA from the crime scene and they analyzed the DNA from Josiah Sutton, they said that there was a 1 in 694,000 chance that this wasn't him. Okay, so basically that another person would have that exact same DNA as 1 in 694,000. That's pretty significant. So he was convicted off of this, but he always claimed his innocence. He said, I didn't do it. I didn't do it. I don't know why it matches, but I didn't do it. And finally, somebody listened to him. Outside company listened, and lawyer took his case, and they went to a different private company, and the company came back and said, yes, that's the case for that section of DNA that they chose, but that section of DNA that they chose to use at the Houston Crime Lab was actually very common in Black men, and it really would match 1 in 16 Black men. That's, if you think about those odds, that's like if you walk down the street and say you have, or, an apartment complex or something, say you have one man in every house, that's 1 in 16. Now you add 2 or 3 men to a house or whatever, or individuals to a house, and that becomes, every other house or something, or every other apartment or whatever. That is horrifying that that was actually used for that and was actually able to convict him. This outside company that did this came back and used other DNA and found out that he didn't match the DNA when you use that. This is why I stress that it is really, really important that they use DNA that is specific to individuals and not specific to humans as a group, or ethnic groups or anything like that, because we all inherit certain traits, and we inherit certain traits through our ancestry. So certain sections of DNA are going to be similar across family lines, across group lines, or whatever else. So it's really important to pick out those specific sections of DNA that are completely different from anybody else, those that make you as a unique person. He unfortunately served 4 years out of his 25-year sentence. He was exonerated. He did leave. Houston Crime Lab, I don't know if any of y'all are old enough, but the Houston Crime Lab came under a lot of fire for a good reason for this, because if he was wrongfully convicted, that means that every single case that they looked at could possibly have been errors involved in that. So they fired a bunch of people, and they went through a whole renovation of it. Austin Crime Lab went through that very recently too. It's really important when you're doing that kind of work that everything falls into place and everything is done correctly. It's people's lives that you're putting on the line there, whether it's the person who's mistakenly convicted, as in Josiah Sutton's case, or it's the victims that are not getting the justice that is due to them. Either way, you're talking about you want to have the correct person that is convicted for that. He did get a lot of money out of this, but it took 4 years out of his life that he can't get back. So, there's not really anything that you can generally say about that. So it can be used, as in Tommy Lee Andrews' case, to convict the right person, and it normally is. I'm not saying that it's not, but it can also be used to exonerate an innocent person, and in this case it was. We can also have it, though, in cases that, really doesn't matter what the DNA says. Sometimes the case just works out for this. This one is a pretty famous case in here. You have, again, several different samples. We had 2 victims in this case, and then you had a suspect that was from here. And again, here's the standard, so you kind of have to ignore the standard in there. And then they got a suspect. So they have to compare the suspect against the blood samples that you found at the crime scene, because we know that the blood at the crime scene are going to be the victims, right? But you can also assume that there might be some residual blood or tissues or something that's left behind by the suspect themselves. So I have to rule out what came from the suspect and what came from the victims before you can make a determination if that anything that's left of the crime scene was actually the suspect's. If we don't look at the victim's blood, then you can't eliminate them as having contributed that sample to the crime scene. So here's some of the DNA that was given here. And remember, it doesn't matter how dark or how thick the lines are, it's the location. So these arrows that you see here point out locations of the blood sample. So here's blood sample from the crime scene 1, and you can see the lines here, the lines here, and the lines here. And they actually match up with the suspect right there. Okay, blood sample from crime 2, from no arrows there, so let's look at 3. 3, you've got here, here, and here. So again, that's the same one there. The ones from the crime scene 2 actually match up with one of the victims. So based off of that, you would say, hey, that suspect was there at the scene, but this case was actually in the mid-1990s, and he was not convicted. See there, those 2 match up. He was not convicted of this crime because you can also have very good lawyers that look at things. And from what I've seen, we've just had the 25 years since this case was tried, and O.J. Simpson did not get convicted of this. He did get convicted on civil lawsuit later by the families, but he was not convicted, even though his clearly, you can see matches going on in this crime scene blood. But there was a lot of issues with probably how this case was handled. Remember, again, 1990s, DNA was still fairly new, and so people didn't understand it, and two, manipulation and stuff. I already told you that crime labs can make mistakes, and so sometimes. And I'm not saying crime labs make mistakes on purpose, obviously, but they can make mistakes. So that is something that you need to look at, not as your sole evidence, but as you have to have other evidence that also would convict the individual. All right, so let's look at structure of DNA from the uses, forensically, to actual structure. What is it that makes DNA so important? First of all, we learned about nucleotides. And here you have a nucleotide. A nucleotide is made up of phosphate, a sugar, and in DNA that's deoxyribose sugar, and one of 4 different nitrogen bases. Okay, and they're all covalently bonded together. So that one section there in that picture is a complete nucleotide. Phosphate, sugar, deoxyribose for DNA. That's where the D in DNA comes in. And then one of, like I said, the 4 nitrogen bases. It can be adenine, it can be thymine, it can be cytosine, or it can be guanine. Every single living organism, from bacteria to an amoeba, protist, to plant, fungus, and animal, all of our DNA is made up of the exact same thing. 4 different nucleotides, and the only thing different about these nucleotides are those 4 things there: the adenine, the thymine, the cytosine, and the guanine. It's a really, really simple method of coding for things, but it's really, really, really effective method too, because thousands and thousands and thousands of nitrogen bases difference can make for different genes. Okay, it's universal, everything uses it, but it's very distinctive too. For humans, for instance, there's 3.2 billion copies of this in our genome. There's 24 different types of chromosomes. Earlier I told you there were 23 pairs for each individual. Remember that if I got one from mom, I got an exact copy from dad. And that's going to go along, for all of your chromosomes. So you have some that are very long, some that are much shorter. The difference in that 24th copy comes from our last set of chromosomes. So we have the ability to either inherit 2 X's, an X from mom and an X from dad, that makes you female, or we can inherit an X from one parent, that would be mom, and the Y from dad, which is much smaller, by the way, and that would make a male individual out of that. The Y chromosome itself is really just about male features. So everybody has X's, so X's don't determine gender at all. By default, in most vertebrates, including humans, or in most mammals, most vertebrates, including humans, the X, well, let's just go with mammals on the thought. The X does not determine gender. The default is just to be female. You inherit an X, and that's all you inherit, you're going to be female. It takes the Y chromosome right there to actually determine gender. And what that says is, instead of going to default female, we're going to go and make this baby a different direction. We're going to make this baby a male. So if you don't have a Y, you're female, period. Remember, men all have X's, so X's doesn't have anything to do with feminine traits. The Y is what determines gender in humans. Other vertebrates use a lot of vertebrates use this method of gender determination, but some things are different, like in birds. If you get 2 of the same, and we usually refer to it as Z's, if you get 2 of the same, you're actually male, and if you get 2 different, you're female. So there's different methods of gender determination, In reptiles, a lot of time it has to do, gender determination is simply by the temperature of the soil. So they're all born with the same chromosomes, but if the soil temperature is a little warmer, they're going to be one gender, male or female, I can't remember. And then if it's a little bit cooler, they'll be primarily a different gender. So there's different methods of gender determination, but in humans, it comes down to XX is female and XY is male determination. All right, structure of DNA. Remember, you have the nucleotides, phosphate, sugar, nitrogen base. We have two sort of setups on these nitrogen bases. Since the phosphate and the sugar are exactly the same for every living thing on the planet, the only thing that's different is going to be the combination of our nitrogen bases. That's what determines the actual code that we're going to have in DNA. And we can have different chemicals. You can have what we call a two-ring structure. And if you look here, this is a ring and this is a ring, and they're bound together. That's a two-ring structure. This one is a ring and that's a ring. That's a two-ring structure. These two-ring structures are referred to as purines. All right. And two of our nitrogen bases fall in this group, adenine and guanine. And then we have this other group called pyrimidines that only have a single ring. So here's the single ring. Notice they all have stuff coming off of them, hydrogens, carbons, whatever, but it's that central base that we're talking about. So purines are a double ring structure and pyrimidines are a single ring structure, and that includes the bases thymine and cytosine. So now we've categorized them into two groups. And what we have found, and we found this out in the late 1940s, is that it is always one purine and one pyrimidine that bond together, okay? So adenine and thymine are found paired up, and guanine and cytosine are found paired up, always as groupings, okay? And this is universal, every single organism. It's not just in humans. This is everything. When you're trying to remember which two are purines and which two are pyrimidines, there's a little sort of a mnemonic device, a little memory device. Some of you may like it, some of you may hate it, depends on which colleges you're affiliated with. But adenine and guanine, Ags are pure. I happen to be an Aggie, so that works for me. Ags are pure, and everything else is a pyrimidine. So that's up to you. But I even heard a student one year from UT who had learned that particular mnemonic too. She wasn't real pleased with it, but she learned it. All right, so what we have found, like I said, is it is always one purine with one pyrimidine, and it's always a specific purine with a specific pyrimidine. Adenine and thymine, and you could just refer to them as A and T's. You don't have to say the whole words. But adenine and thymine always pair together, and cytosine and guanine always pair together. This was determined in the 1940s by a scientist named Chargaff. And so it's sometimes referred to, you'll hear it as Chargaff's rules. Other times you'll just hear it as base pairing rules because it's the way the bases pair together. All right, those rules. And it was just discovered by percentages. All he did was a chemical analysis and say, ooh, adenine and thymine always occur in the same amount. If I have 30% adenine in my sample that I'm chemically analyzing, what we find out is there's about 30% thymine also in that sample. If I have 20% cytosine, then I'm always going to have 20% guanine. So these equal and these equal. And that's how we determine that A and T were always found together in C and G. By the way, you can always figure out how much of each one there is too. So if I tell you that there's 30% adenine, you automatically can figure out there's 30% thymine, right? Well, 30 and 30 is 60. Percentage-wise, that only leaves 40. And since these have to be equal, you divide that in half and you can get those two. So we can also determine quantities of each one. So then what we have from this is a polynucleotide that is formed. So right here, I have one nucleotide. And then next to it, I have bonded to it a second nucleotide. And then if you look on the other side, I'm going to make a circle on that one. There's another nucleotide, and there's a next. And then they just bond together in these lines along here. Now in the center, so this is all covalently bonded right here. Each nucleotide is covalently bonded, and then is covalently bonded to the next one on down the line like this, okay? And the same thing here, covalently bonded this way. In the center is where we see a difference. Between those nitrogen bases, where I said that these pair up, A to T, T to A, doesn't matter which direction, it's A and T or T and A, C and G and C. Right here, you see these dotted lines. Those are hydrogen bonds, okay? Hydrogen bonds, if you remember back from chapter 2, are weak bonds, but they're still bonds. So they hold one half of the ladder together. So this half is being held to this half with those hydrogen bonds. And this is why you always see A and T together and C and G together. If you notice, pay attention real quick, A and T can only form two bonds. You see it here, right? And we see it here. They form two bonds. C and G like to form three bonds. So they have to. So since C wants to form three bonds and G wants to form three hydrogen bonds, they can only match up. You can have 3 and 3, right? But you can't have 2 and 3 because then, somebody's left without a bonding partner, right? So you have to have 3 together or 2 together. It matches up. That's the chemical structure. All right. And like I said, then that forms these long strands. You can see them in blue of what we call a polynucleotide. So each one of these is one nucleotide. Many of them is a polynucleotide. And in DNA, you have 2 strands. So we have 2 strands that are have hydrogen bonds between the A's and the T's and the C's and the G's, right? So it's a double-stranded nucleotide. They do run opposite directions. So this one runs this way and this one runs this way. Parallel, but anti-parallel because they're running opposite directions from each other. Notice here, when I say opposite directions, see the phosphate right here? The phosphate indicates my first nucleotide, but on this end, the phosphate's down here. So it's actually running from that direction up. So that's why they're opposite. Okay, dog's over there snoring. Okay, hydrogen bonds then are going to hold those nitrogen bases together. That's the A to T with 2 hydrogen bonds and the C to G with 3 hydrogen bonds, right? And that goes down the center. And then that starts to twist up. So it twists up into what we call a helix, and we call it a double helix because there's 2 sides to it. In eukaryotes, which are organisms that have nuclei in their cells, so the DNA is actually located in a nucleus. Our DNA is so long, 3.2 million base pairs, remember that, on just 23 different types of chromosomes. Or 24 types, so 23 for each individual. We have so much of it that we have to organize it. So we organize it by wrapping it around little proteins called histones. So this that you see in this picture is DNA. When it starts wrapping around those little proteins called histones, then it becomes what we refer to as chromatin or chromosomes when it shortens and thickens during mitosis. So chromatin and chromosomes really aren't DNA by themselves. They're DNA plus protein component. And then that is forming our chromosome that we're used to seeing. All right, so that's the structure. Now we want to talk about how it holds the code. Remember, the key is in the only thing that's different, and that would be your nitrogen bases. What order do I have this A, T, C, G? And just because I have an A doesn't mean I have to have a C next. If I look down one side, I could have multiple A's in a row before I threw in something different, right? So A's, T's, whatever. There's so many different ways of putting those 4 letters in an order, because they don't have to be different each step, they can be the same, that causes the differences between you and a bacterium. All right. Every time you started out, when you started out, you were an egg, and there was a little sperm that came in, and then that gave you. So mom had 23 chromosomes, dad's sperm had 23 chromosomes. When that entered, that gave you 46, and we call that a zygote. Okay, a zygote is just a fertilized egg cell. Immediately, once this occurs, that cell starts to go through mitosis, and you create 2 cells. And then those divide, and you create 4 cells, and so on. And it continues to divide very rapidly till you're a ball of cells. And then, like I said, those cells will start becoming an embryo. An embryo will become a fetus, a baby, a child, an adult, and so on down the line. Every single time that one cell divides into 2 cells, you have to copy your DNA. That's called replication. So your 46 chromosomes have to actually copy into 96 so that I can get 46 and 46 into each cell. If you don't do that, then within one replication, you would go from 46 to 23, right? And then the next replication, 23 and a half is 11 and a half. You're starting to break up your DNA at this point, into each cell. And then 6 and a quarter, 7 and a quarter, I don't remember, 6 and a quarter, or 6.75. You're breaking up your DNA, and it wouldn't take very many divisions before you just had just pieces of DNA and nothing left. So once again, we have to copy first and then divide down into 2. That is called replication. So I have to make a copy of my recipe book and then put a copy of my recipe book into each new cell. So to do that, since our DNA is all wound up, first step is to unwind it. You have enzymes, by the way, that control each of these actions. So you unwind your DNA, you unzip it, and that really just means breaking the hydrogen bond. So here, where you see this bubble, what we call a replication bubble, these bonds between the nucleotides or the nitrogen bases have broken to allow for new DNA to come in there. All right. So unwind, unzip, second step, and then bring in free nucleotides. So free nucleotide is just a nucleotide that's not bound with anything else. So if I have this down at the bottom, here's my original ladder. We skipped the unwind, but it unwound. Step 2, it's unzipping, right? So this is unwinding, unzipping. And now my A is exposed. The first nucleotide right here is exposed. That A does not like to not be bound with something. So a nucleotide that's roaming around the nucleus all by itself will come in and bind with it. And since A likes to bind with T, it would be a T that bound here. On the other side, likewise, you have a T that's exposed. So an A nucleotide will come in and bind in. C with G, T to A to T, G to C, G to C, A to T, T to A, and G to C. And what you end up with at the end are 2 identical copies. Notice that this strand here contains the original ACTAG from my strand I started out with down here. And then this one contains the other side that was original. So each new strand contains an original side plus a new side that was built using that code that it already had on there. And then if you compare these two, they're exactly the same. Now this strand can go to one cell and this strand can go to a second cell, and we've just made an exact copy. And you make a copy of every single DNA using that. There's a bunch of enzymes that are involved. I want you to know this one. DNA polymerase is the enzyme that allows this process to occur. All right, copy your DNA. And it does so almost without any flaws, right? What happens if you had a flaw right here? See this C? What if I brought in, let's drop down a little bit. See this A right here? What if instead of bringing in a new T, I brought in a C right there? This would be an incorrect code. It doesn't match this other side over here. This would be an incorrect code. You do that. But DNA polymerase not only brings in the right nucleotide at the right spot, it will check for that and it'll say, hey, that was wrong. Let's correct that and bring in the right thing. So it cuts out the incorrect and brings in the new ones if a mistake occurs. Because a mistake that goes unchecked is a mutation. A mutation means it codes for a different protein. And if I change my protein, I change my amino acid sequence, right? Then I change the function. So if I bring in the wrong amino acid at the wrong spot, I make a different protein, I don't get what I need for my cell to do the cellular work. So that would be, something you want to limit. All right, so let's look at this process occurring. So I have my original strand. It's already broken apart, so it's already separated and unzipped, right? Here's a nucleotide right here. Likewise, this was the nucleotide on this side that bonded across. So G and C right here and right here bonded together, A and T across bonded together, and so on. This is my code going downward. So when you look at the code, you look at G, A, G, T, C. So we always look straight down one side of the ladder. Or likewise, I can go this way, G, A, C, T, G on that side, okay? To replicate, the next thing that has to happen is you have to bring in a nucleotide. So a free nucleotide, stop. Sorry, it's going ahead of me. A free nucleotide first comes in. That is just a phosphate of sugar and a nitrogen base. And since this is C on this side, it has to be a G that bonds in here. Since this was T on this side, you have a second free nucleotide come in that is the adenine nucleotide, and it bonds with the T, C to G, A to T, G to C. Very, very simple, but very, very effective, because now I've got this side here actually matches this one over here. So they're exactly the same. Likewise, would do the other side, but here's the problem. Those phosphates are energy, so it has to build the other direction. And there's another enzyme involved. So we build one side going down this direction. We build the other side going the other direction. All right, that's that anti-parallel thing. But notice the important part here, our code all the way down has remained the same on both sides. So now I have an exact copy of what my original was. This can go to cell number 1. This can go to cell number 2 during mitosis. All right, so this is what's happening in your cells all the time. In 1985, this guy that was a surfer at UC Berkeley, he was a grad student and a surfer. That's why you see him with the surfboard in his spare time. He was trying to figure out how we can make this replication thing happen outside of a cell. How can we take a piece of DNA and make a copy of it? Your cells do it all the time. Your cells make copies of DNA, so why can't we artificially do that? And the legend goes, or at least his story goes, that he was on his way to go out surfing. That's why you see him with the surfboard all the time. And it kind of hit him. Well, what do I need to do? I need to take the DNA that's wound up. I need to unwind it. We need to open it up. And then we need to bring in the free nucleotides. And you got to have all the enzymes that are involved in that. So he said, all we really have to do is basically provide a fake cell for this to happen and then provide all the ingredients. So he figured out, how do I get the DNA apart? I just heat it up a little bit. I break those hydrogen bonds. So he developed this technique that we call PCR, and it won him the Nobel Prize in 1993. And it's like, well, why was that so important? PCR stands for this term, polymerase chain reaction. Without him developing this, we could not have made all the strides in analyzing DNA, in forensics, in genetic disorders, trying to cure genetic disorders, and all the things that we have learned since then is basically due to this artificial DNA copier that he was able to replicate or make in his lab in order to make DNA. So what we can do is, because of him and because of this advancement that he made, we can have only one cell that's been left somewhere, one cell of a person's DNA, and we can make millions and millions of copies of that DNA and be able to analyze it without destroying the DNA. So before, like in the 80s and early 90s and stuff, you had to have a large sample of DNA that was left behind so that they could take part of that DNA, analyze it, and keep some intact. Now we just need one cell, and he can, with his little machine, make millions and millions of copies, and you've got, exact replicates of that original DNA. So too little DNA to work with. All you need is a small amount. All you need really is one cell. And you just take that one original piece of DNA, do what the cell does, but do it in an artificial environment in what we call a thermocycler. A thermocycler is a machine that does PCR, okay? Take the DNA, split it apart. So that's step one, split it apart. Add all of the little pieces that you're going to need to get the DNA to make copies. So we have these little things we call primers that are in that mix, in that artificial cell, that PCR, that thermocycler, and then build 2 new ladders. So we went from 1 ladder to 2 ladders right there. And that takes about 30 minutes now. I don't know what it took him back in the day, but it takes 30 minutes now. Then repeat the cycle, cycle 2, do all of those steps again, and we go from 2 to 4 in another 30 minutes. And then we can go from 4 to 8, and it becomes exponential. So within a short period of time, I can have my kids do it, and within a couple hours, a few hours, actually, not really a couple, but a few hours, we go from a very small amount of DNA to millions of DNA strands that are all exactly copies of each other. And it just takes cycling, replication cycles to do that. And because it is an exponential factor, so I start out with very little, if this is a graph, very little, it becomes a very sharp upturn in how much DNA there is in a short period of time. And then you can analyze it. Then you have enough to work with. So somebody can leave behind one cell at a crime scene, or a body that has degraded too much and only has a few cells to work with for identification purposes. You can take those few cells, and we can make millions of copies, and then we can analyze it. Or if you're going to do something like 23andMe or Ancestry or something, one of those, you can just get a few cells from your cheek with the spitting process that you spit into the tube. Instead of giving them a whole chunk of cells, you can give them a few cells, and they can go through this PCR process and then be able to analyze it and break down your ancestral history, your genetic history for diseases, if that's what you want to know. 23andMe does that, and see, what you've inherited. So all we need is a PCR. Here's one I just took the picture off the internet. This has gotten so cheap, or relatively cheap enough, that I actually have a PCR in my high school classroom, and my high school kids can do this whole process. So we went from, only in science labs to, research labs to high schools where it's available. It's not, on the level of something you would find in a research lab, but it's a PCR that works. So it's good enough for what we need in classroom. You have to have a template strand. So that will be the DNA that you're starting with, that you're working with, that you want to make copies of. You have to have DNA polymerase, because remember, DNA polymerase is the enzyme that makes the copying occur. You have to have free nucleotides. So I have to have running around A, T, C's, and G's bound to their sugars and phosphates, but just those separate nucleotides by themselves, and throw that into the mix. And then I have to have some primers and other enzymes that, I kind of skipped over the other enzymes that will allow the nucleotides here to bind to the template and make new strands every time. And then repeat, put all that together in a tube, repeating, heating up to break apart the hydrogen bonds, cooling to allow the two sides of the ladder to build, and then extension of those ladders, and then repeat through this over and over again, go through that cycle, and I can make millions of strands. It's like a copy machine for DNA. And then all you have to do is take this big bunch of DNA and take a section out and study that and keep the rest for use later if necessary. And that's going to be all for replication. So remember, replication itself is going to be unwinding, unzipping, and then bringing in the free nucleotides. And it simply is for the process of making new copies of DNA to put in these cells.